Let's talk about protein structures. Protein structures have 4 levels. The first level structure called the primary structure. Primary structure is a polypeptide chain with a particular sequence of amino acids. And this here you can see an example. It's the chain made of amino acid residues. It's a polymer. On the left is the N-terminus, on the right is the C-terminus. You can see the monomers on the blocks. The methionine, valine, glutamic acid, alanine, serine, isoleucine, leucine. This is the primary structure. The next level is called a secondary structure. Secondary structures are three-dimensional arrangements of segments of a polypeptide. They're formed by hydrogen bonds between amine groups and the carbonyl groups of amino acid residues. There are 2 types secondary structures. One is called alpha helix. Alpha helix is a right-handed spiral. To be an alpha helix, they must meet the following structural requirements. The first one, each turn of the helix has 3.6 amino acid residues. The second one, the carbonyl of one amino acid residue is hydrogen bonded with the amine group. 4 amino acid residues farther along the peptide chain. And the third one is the R groups of the amino acid residues extend outwards from the helix. Here is you can see a model. And first, you can see the turn. This in general, this is alpha. This is the right-handed spiral. You can see each turn of the spiral has 3.6 amino acid residues in on average. Then you can see the hydrogen bond, the force for forming the alpha helix, the hydrogen bond. You can see this dashed line represent the dashed lines represent hydrogen bonds. You can see this is the carbonyl group, is the amine group, and between the 2 residues, the 3 amino acid residues has 3 amino acid residues apart from this one and the one which forming the hydrogen bond. Then next you can see the R groups. Okay, it's the R groups extend outwards from the helix. The second secondary structure, second type of secondary structure is called beta sheets. Beta sheets, 2 or more beta strands of peptide fragments line up side by side to form a beta sheet. Beta sheet has the following features. The first one, all the hydrogen bonds formed between carbonyl group and amine groups line up on the plate of the beta sheet. Hydrogen bonds are formed between carbonyl and amine groups between 2 adjacent strands. The R groups of the amino acid residues are oriented above or below the plate of the beta sheet. This is the model here. You can see this is the beta sheet made of 4 strands. 1, 2, 3, 4, and you can see those strands can be anti-parallel. The first one, the second one, second one, the third one, all parallel. Okay, the third one and the fourth one on the same direction. You can see the hydrogen bonds between the carbonyl group and the amine group is red colored for the antiparallel strands, and the green colored hydrogen bonds on the parallel strands with the same direction. Those hydrogen bonds on the plates on the same level of the plate of the beta sheet, and the R groups oriented either above the sheet or below the sheet. This is the secondary structure. The next level is called the tertiary structure. Tertiary structure is a three-dimensional structure folded by the entire single polypeptide chain. Generally, hydrophilic groups are on the surface of the protein to interact with the water molecules and ions in the aqueous environment. Hydrophobic groups form a core of tertiary structure of the protein van der Waals interactions. In addition, salt bridges, hydrogen bonds, and disulfide bonds are also formed for maintaining stability of the structure. This is the model for the tertiary structure. It's a lysozyme. You can see the alpha helix and the beta strands, two beta strands, beta sheets. This is this structure formed by entire polypeptide chain. This is a single polypeptide chain. And the last level is called the quaternary structure. This structure is formed by two or more polypeptides. Each polypeptide is called the subunits. This is the model here. It's a hemoglobin. Hemoglobin is a tetramer formed by 4 polypeptides, 4 subunits. And in this module, you can see 4 colors. Each color represents 1 subunits.